Today, I'll give an introduction to the Gibco OncoPro Tumoroid Culture Medium Kit. Cancer is a major public health problem worldwide, leading to around 10 million deaths annually. Fortunately, the drug development process for new cancer drugs is quite lengthy, often taking around 10 years and very expensive, averaging around $1 billion to develop a new cancer drug. Some of this cost is reflected in the fact that around 95% of cancer drugs fail in clinical trials. To address this issue, many researchers have turned to more complex cancer models. Traditionally, cancer models have been used during functional testing of new therapeutics to assay and check the response of cancer cells to new compounds or therapies. This is often done using immortalized 2D cancer cell lines, which can also be grown in 3D as cancer spheroids. These immortalized cell lines, unfortunately, are often hypermutated and have limited complexity, not really reflecting the heterogeneity of human cancers. In many ways, this leads to their low clinical relevance, where promising in vitro results often don't translate to the clinic. There's a number of ways that these models can be made more physiologically relevant, which often comes with increased complexity as well. Primary cells freshly taken from human cancers can be grown in 2D as 2D primary cell cultures, in 3D as cancer spheroids when these 2D immortalized cell lines are forced to aggregate into a 3D shape, or potentially as cancer organoids or tumoroids, which I'll talk more about here. Many of these cell models can also be incorporated into organ-on-a-chip devices to bring in further complexity, geometrical organization, or other cell types. And during the development process, these are typically then moved into animal models before going into clinical trials. The downside of looking at many of these models is that they are more costly, take more time to set up, and have lower throughput than fairly simplified assays using 2D immortalized cell lines. Today we'll focus on patient-derived tumoroids, which we feel are a really nice way to get some of the benefits of these highly complex cancer models while still enabling some of the throughput that traditional in vitro assays allow. Patient-derived tumoroids are more physiologically relevant and show improved in vitro predictivity for what will happen in clinical settings. This brings the biological relevance of these cell models upstream with the goal of failing early and failing cheaply during the drug development process to hopefully lower the cost and improve the efficiency of cells moving from the lab to the clinic. Tumoroids are also known as tumoroid lines, cancer organoids, or PDTOs, or patient-derived tumor organoids. These are patient-derived cancer cells that are grown as 3D self-organized multicellular structures. Complex cancer models, and they come with complex cell culture requirements as well. Interest in these models is really driven by the recognition that immortalized cell lines don't look like patient cancers. So on the left of this slide, we've got some data that we generated internally to just show that if you randomly look at a tumoroid line, a patient cancer tissue sample, or a cell line, these are all colorectal cancer cells in this case, what you see is that these immortalized cell lines are often hypermutated in terms of having higher tumor mutation burden than clinical tissues or tumoroid lines and more clinically relevant mutations. Additionally, gene expression patterns of these 2D cell lines are quite distinct from patient tissue and from tumoroid lines, as you can see from the heat map in the middle of this slide. As these tumoroid models better reflect patient cancers, there's been greater and greater interest in the research community for using these 3D models. You can see by the increasing number of PubMed search results for both organoids and cancer organoids. In our minds, these tumoroids can really be used anywhere that a traditional cell line would be used during cancer research. This can be in basic research to look at mechanisms of cancer development, in drug discovery and biomarker identification pipelines, as well as for development of immuno-oncologies and eventually companion diagnostics. So if these are such great models, why aren't they being used everywhere? We feel that a lot of the the limited use of these models thus far is due to some challenges with the current methods for tumoroid culture. If you look in the literature, these models typically require quite complex media that was originally developed for healthy stem cell-derived organoids. Most of these recipes require 10 plus components, typically require either conditioned media or recombinant proteins, and are not grown with a standardized formulation. In fact, if you look through the publications, many different labs will have their own unique homebrew formulation for growing these models. Traditional methods are often quite labor intensive as well, requiring the use of matrix embedded hydrogel domes for culture that does really well at preserving the 3D architecture of these structures, but is not particularly scalable or automation friendly. Finally, there are some real IP restrictions in terms of methods that can be used to propagate these cancer organoid models. And it's often needed to have a license to use these cells, the media, and the methods involved. On the right is showing an image of what these Geltrex or basement membrane extract domes look like. These are 3D matrices that are often spotted one at a time onto a cell culture dish, and then have to be broken up to free the cells for either downstream assays or even just for routine passaging, which is a fairly time-intensive process. So that led us to develop the Gibco OncoPro tumoroid culture medium kit. Our goal here was really to simplify tumoroid culture to make these physiologically relevant cancer models more accessible and more standardized for use throughout the cancer research community. This image on this slide shows what this OncoPro medium kit looks like. There's a basal media and then 3 supplements that are all kitted in a 500 milliliter size. And this media has been optimized for epithelial-derived carcinoma. Most of our the indications that we have the most data with are with colorectal, lung, head and neck, pancreatic, breast, and endometrial cancers, although we anticipate that this media system will be compatible with other epithelial-derived carcinomas as well. I'll show some data here to demonstrate how this kit has been optimized for patient-derived tumoroid cultures, how we have worked to streamline media preparation and culture methods, and how it's adaptable for multiple cancer indications and downstream assays. So let's start talking about optimization of this media system. OncoPro Medium is a de novo medium designed for patient-derived tumoroids, where we used a number of screening approaches to identify conditions that promoted cell expansion and survival of these 3D patient-derived models, while also, as I'll show you later, maintaining the donor-specific characteristics of these cells. So on the left, you can see an image kind of showing a screen that we did where we see some conditions giving very good performance and robust tumoroid formation, while others showing very low cell growth. We've looked at that by imaging, by other cell viability assays across a wide number of media conditions, and really focusing on identifying key supplements and factors that were beneficial to tumoroid growth and propagation. Those components were then selected and titrated to find concentrations that worked as broadly as possible across as many cancer models as we could use. This system was really designed for the expansion of cancer cells in the absence of Wnt, noggin, R-spondin, and small molecule inhibitors. What we see over time as we characterize these cultures is that uncultured samples, which may contain blood cells, stromal cells, other non-cancer cells with our beginning material, are eventually selected to grow tumoroid lines that are enriched for cancer cells and typically have greater than 90% EbChem expression after several passages and after several months in culture. The plot on the bottom left shows an example in colorectal cancer where we see selection for cells that are EbCam and CCAM positive. CCAM is a marker for colorectal cancer. And we're losing expression of CD45 and CD31 as immune and endothelial cells die off over time. Importantly, a number of the tumor lines that we have derived retain tumorigenicity and can be implanted into immunodeficient mice to grow patient-derived tumoroid xenograft or PDTX cultures. So the plot here shows tumor volume in a mouse over time after these tumoroid cells were implanted, showing that these tumoroids begin to form and reach large sizes in vivo in a mouse model. Interestingly, these models retain similar morphologies and staining patterns, both in vitro and in the PDX tumors. This all occurs in the absence of Wnt signaling. So if we look at Wnt activity in the media, again, several positive controls, including homebrew media recipes that we know activate the Wnt pathway. You see that OncoPro media is not activating this pathway, but importantly is also not leading to changes in the profile of the cells that are grown when Wnt is not activated. So for example, if we culture pancreatic cancer tumoroid models that were established and grown in a media that contains Wnt activators, and then transition them to grow in OncoPro, which does not have promoters of Wnt signaling, we see very, very few genes that are differentially regulated between those pairs. Most importantly, these OncoPro tumoroids are representative of the original tumors from which they were derived. So for much of our workflow during development, we took fresh tumor resection samples, associated those, and then characterized the starting material or cultured and expanded cells in OncoPro media to establish stable tumoroid models. These established tumoroid lines have different morphologies, which are quite diverse, as you can see from these images on the slide, and really are donor dependent. Some examples here are shown for colorectal, endometrial, and lung cancer. Many of our experiments were aimed to establish stable tumoroid lines, which we defined as cell lines that could be split every 14 days or less for 5+ passages and expanded for more than 5 cumulative population doublings over the initial cell material that we had. They also had to maintain patient-specific characteristics and be freeze-thaw competent, that they could really be used like you would with a traditional cell line. When we talk about these cells maintaining donor characteristics, we looked at that largely by doing both mutational and gene expression profiling. So on the left, you can see plots for a number of different cancer indications: colorectal, lung, breast, and endometrial, looking at oncogenic mutations and comparing for each of these pairs of two, the original material on the left to the established tumoroid line on the right, and showing that the same oncogenic mutations were present in both cases. Also worth noting that we've seen a wide variety of mutational profiles supported in the media system. So for example, not all cancers that we can grow are p53 mutants, not all are BRAF mutants, but instead we're really reflecting a wide variety of genetic backgrounds. We see similar patterns for gene expression profiles, where we can compare the level of gene expression in the initial tumor cells and the derived tumoroid models, again, across a number of indications, and show very high concordance and high correlation of the initial material and the resulting tumoroid line. In fact, this correlation coefficient between those two comparisons is typically above 0.8 once these stable lines are established. Importantly for research use, these donor-specific characteristics are maintained over the long term. So here we're showing cells comparing an initial bank of tumoroid lines derived from a patient tumor material, cells that have been cultured for a long period of time, often more than 30 passages, to demonstrate that the morphology of these lines are maintained over these months in culture, and that the growth rates are also stable over time, including after freeze-thaw cycles. So the middle plot shows, in many cases, over 250 days of growth, including a freeze-thaw cycle for colorectal and lung tumoroid lines, show that this growth rate and population doubling rate is maintained over many passages in culture. Importantly, this occurs while we're maintaining individual gene expression patterns. I'm showing two examples here of colorectal cancer lines that are comparing an initial bank at passage 13 to banks at passage 50 after culture for many months in culture. And you can see that these indication-specific subtypes are maintained in the cultures. So Huco 021320 is largely goblet-like and inflammatory, which are two phenotypic subtypes of colorectal cancer, and those are maintained over time, while Huco 3209 is much more transit amplifying. And again, that expression status maintained all the way from passage 13 to passage 50. There's other ways of looking at that. So that was from a gene expression level. We can do a similar study looking at mutational profilings. So the plot on the left is showing the allelic frequency of different single nucleotide variants, or SNVs, over, in many cases, 250 plus days in culture. This is a correlation plot comparing our initial bank at passage 13 or passage 4 to 5 with passages much later in time, often up to passage 50, to show that not only are the same single nucleotide variants present in a donor-specific manner, but they're there at similar frequencies. We also see very little cross-correlation between different donors, so that you can see that these are not driven to a common mutational profile in the media system, but are instead maintained in a donor-specific manner for several colorectal and lung cancer models in this example. We see a similar thing for gene expression profiles. So this is a PCA, or principal component analysis plot, comparing 3 colorectal donors and 2 lung cancer donors to show that these gene expression profiles cluster by donor over multiple passages in vitro up to, again, passage 50 in many cases, instead of converging on any common phenotype as they're cultured. So overall, the donor-specific characteristics of these cells are maintained over the long term when they're grown in OncoPro tumoroid culture medium. So next, let's talk about streamlined media preparation and culture methods that go along with this approach. OncoPro medium represents a simplified media system that was designed for these tumoroid cultures. As I mentioned previously, it is an off-the-shelf 4-component kit that is adaptable to support multiple indications, sometimes with the addition of other growth factors to support specific niche signaling pathways, which I'll get to in a minute. It's cost and time effective and reproducibly manufactured so that it can be used as a standardized approach across time and across labs to grow these physiologically relevant tumoroid models. Also, no license required for commercial use of this media system. In comparison, a lot of these homebrew formulations require extensive validation, tailoring as they move from one lab to the other, and as they need to be validated with the individual components, often individually aliquoted and recombined to make media. In many cases, these are very complex formulations requiring 10 or more components from multiple vendors that have to be individually handled and stored as you're making your media. We'll get into some of the time and cost that is involved with this, but the lack of standardization in our minds with this approach has made it harder to, one, be adopted by new labs, and then two, to have good comparison of data from one system to the other. One advantage of OncoPro media is that we have quite a bit of data showing that it is consistent and reproducible in terms of lot-to-lot reproducibility as users use this to propagate their tumoroid lines. So on the left, we can show that and have seen that there's consistent doubling time for both non-small cell lung cancer and colorectal cancer lines across multiple lots of OncoPro. So we're seeing essentially the same growth rate of these as we use different lots of the media. And perhaps more importantly, we're also seeing very similar gene expression patterns, again, across both lung and colorectal cancer models here, as we go from one lot of OncoPro to the other. So this is really important in terms of just the background status of tumoroid models being maintained consistently from one lot so that these experiments can be standardized and can be done reproducibly with minimal background variability in the status of the cells. We feel that OncoPro is more cost-effective than these homebrew or DIY approaches as well. So on the left, we've got a plot of the cost per 500 milliliters of media plus growth factors that are needed. You can see that OncoPro medium, depending on any additional indication-specific growth factors that need to be added, costs between $671 and $764 per 500 milliliters, which is significantly less than the average cost of homebrew media systems using recombinant growth factors and comparable to homebrew media systems using conditioned media. That's the material cost of the media needed. We've also done a number of experiments actually looking at empirically growing 10 million tumoroid cells from an initial cell vial using these different media systems in both suspension and embedded approaches, as I'll get to a little bit more in a minute. And what we see is that across indications and taking into account, importantly, any growth rate differences that we see from one media system to the other, OncoPro medium is quite comparable to conditioned media homebrew approaches and quite a bit less expensive than homebrew approaches using recombinant media. If you grow these cells in suspension culture, you also save significantly on basement membrane extract costs and can have savings of up to about 30% when you take into account all of the growth factors plates, supplements, and basement membrane extract needed to expand cells and stably culture these lines with OncoPro compared to other approaches. With these cost savings come a fairly significant decrease in complexity as well. So here we've got listed the materials needed to culture cells in OncoPro, either in a suspension or an embedded protocol, and the costs associated with those based on the different growth rates that users may see with different tumoroid lines. A number of the potential cost savings with a conditioned homebrew media in terms of just the base cost of reagents is offset by the fact that before you even get to this point, many of these protocols require conditioned media produced by specialty cell lines that have been engineered to produce Wnt, R-spondin, and noggin. One of the issues here, in addition to the time required to both establish banks of cells to make conditioned media and then to actually produce the conditioned media before making tumoroid media in the first place, is that there's also batch-to-batch variability and need to do characterization on the front end before you even get to tumoroid culture. A lot of that variability can be offset by using instead recombinant growth factors in homebrew media recipes. But that approach significantly increases the cost of the media. And when that comparison is made, you can really see the benefits of using OncoPro, both from not having to aliquot each of these individual components in the homebrew media recipes and not having to separately purchase some of these expensive recombinant proteins. So I've been alluding to suspension culture before. So what do we mean by that in terms of OncoPro? One of the things that we wanted to do during development was make a media system that was compatible with a suspension culture that looks more like traditional cell culture that many cancer researchers are familiar with. So in this suspension culture approach, cells are resuspended in media in the presence of 2% basement membrane extract instead of being embedded in these basement membrane extract domes. This can work for a number of models, both from tumor resections as well as existing tumoroid lines or patient-derived xenograft organoid lines, where they handle much like a suspension cell line. so they can be grown in flasks for scale up and then harvested for downstream assays or banked. OncoPro is also fully compatible with embedded culture approaches as well. So for applications where users are interested in keeping their cells embedded in a 3D matrix, OncoPro can be plugged into those as well. Again, the streamlined suspension culture really gives you a lot more flexibility in terms of scale up. So they can be grown in T25 flasks, T75 flasks, etc. But they retain the morphology and the gene expression patterns of cells grown fully encapsulated in 3D basement membrane extract domes. The real advantage here comes in splitting time. So because these domes have to be broken up in embedded culture, it's actually quite an extensive process to first free the organoids and then dissociate them for downstream splitting or harvesting. And that time is significantly reduced to about an hour to split 2 million cells in suspension culture. Partially due to this and partially due to the fact that these domes don't have to be individually plated, scaling to 1e⁸ cells can be done in about 10 T75 flasks in suspension culture, using about half as much basement membrane extract as what would be required if this was done in an embedded approach. So overall, the suspension approach is what we use day to day. It's simpler, requires fewer resources, it's quite a bit faster when you're splitting cells, and it's highly scalable depending on downstream needs. One thing that we like to emphasize with OncoPro is that this suspension approach really gives you flexibility. So many users are interested in embedded culture, at least for some applications. So it's worth emphasizing that OncoPro is still compatible with this embedded culture. But for times when suspension culture is helpful, there's the flexibility is there to move right into that. Importantly, the suspension approach does not lead to changes in gene expression patterns. So we have cultured a number of existing tumoroid lines that were established in embedded culture from the National Cancer Institute Patient-Derived Models Repository, or PDMR, and done gene expression profiling after expansion in either embedded or suspension culture conditions. And what we can see is that across, again, a number of indications, these gene expression patterns are again correlated by donor and within a given tumoroid line instead of by the condition in which they were grown. So there really is minimal trade-off in moving from one approach to the other. But you can instead maintain donor-specific gene expression patterns and grow the cells in whatever format is appropriate for the experiment of interest. Finally, OncoPro is adaptable for multiple cancer indications and for downstream assays. So one nice thing about this approach is that this 4-component kit can be supplemented with 1 to 2 indication-specific growth factors to support the culture of tumoroids from diverse cancer origins. So here we're showing example tumoroids from a number of indications and any indication-specific factors that we recommend adding to the OncoPro base kit to support those cancers. So for lung and head and neck cancers, that the addition of FGF10 is recommended. For pancreatic cancer, we typically will add in FGF10 and gastrin to support these cultures. And for breast and endometrial cancers, we'll add in FGF10 and beta-estradiol on top of the base formulation to promote the stable growth of these lines. Additional indications may be possible with some testing, and we encourage any users to contact technical support for advice and starting points for other indications not listed here. We have just reflect that we have not had access to tissue to was done using established tumoroid models from the National Cancer Institute patient-derived models repository, or PDMR, to show that these existing tumoroid lines could be transitioned to OncoPro to continue experiments, but while getting all the benefits of standardization and reproducibility and ease of use that OncoPro offers. So our typical approach for those types of experiments is shown here. We received these tumoroids from the NCI and then grew them in their line-specific homebrew formulations or in OncoPro suspension or embedded culture, and looked at growth rate over time, usually over 6 passages, and demonstrated that OncoPro culture in general gave quite comparable growth rates as the homebrew embedded culture that these lines were originally derived in across a number of indications. Some differences may be observed on a line-to-line basis, but our overall average expectation is that growth in OncoPro in either a suspension or embedded format will on average be quite comparable to growth in the homebrew embedded format that these lines were in before. Importantly, this transition from the original media into OncoPro conserves the patient-specific gene expression profiles, as shown on the left with PCA plots from cells initially received from the NCI and expanded in each of those 3 arms of the study that I showed on the last slide. So we're retaining these patient-specific gene expression patterns, as well as maintaining gene expression mutational profiles. So a similar analysis as, again, to what I was talking about before with these frequencies of single nucleotide variants across a number of models, again, showing retention of these mutations in a donor-specific manner as we change from the initial media to OncoPro embedded or suspension culture. Once these lines are grown, they are adaptable for use in a number of downstream assays. We've tried to use these as much as we can. Any place where someone would use a cancer 2D cell line previously, so immunofluorescence, high content screening, flow cytometry, migration and wound healing, cell signaling, in vivo tumorigenicity, electroporation, lentiviral delivery, drug screening, and immuno-oncology are all compatible with these tumoroid models. Part of this is some proof of concept data that we've generated internally, showing that these cell lines can be used for compound screening workflows with a number of different readouts in a 3D approach instead of the kind of traditional 2D approach. So here, tumoroid lines were dissociated, fed or treated at day 4, and then used with either Presto Blue, high sensitivity reagent, or other cell viability reagents to look at the dose response to different drugs, storosporine and gefitinib, are shown here just to demonstrate that these models respond to these drugs and that changes in cell viability as higher concentrations of these drugs are added can be detected in the background of OncoPro media. Interestingly, we see donor-specific responses to these drugs as well. And you can start to see the value of having large biobanks of colorectal cancer models in this case, or models from other indications, to incorporate kind of the heterogeneity and the background variability in different cancer types to drugs, and really seeing how a larger population responds to those different compounds. We do similar things with potential immuno-oncologies and cell therapies, where here one of our tumoroid lines was engineered to express GFP, so that could be used in a fairly high throughput screening approach with primary natural killer cells to look at the killing efficiency of those natural killer cells to these 3D cancer models. So it was a similar approach as before. Cells were dissociated, and then on day 4, after some tumoroids had formed, natural killer cells were added, and those were monitored over time. You can see on the bottom left images of these GFP expressing cells when NK cells were added. Caspase 37 detection dye was detected by imaging methods or by plate readers, whereas when no NK cells were added, we did not see any recognition of this caspase 37 dye. And on the right, you can see that that can be monitored over time. So tumoroid signal from the GFP constitutive expression increases over time as these cells are grown. When primary NK cells are added, that GFP signal decreases in a dose-dependent manner with a greater increase or greater decrease in GFP intensity as more NK cells are added, and a concomitant increase in the apoptosis signal in this caspase 37 dye that again is higher with a greater ratio of NK cells. So we see this nice dose-dependent response in both primary NK cells and an NK92 natural killer cell line that we can detect using these 3D cancer models. I should mention that there are a number of services available to do this type of work. These tumoroid models can be used with cell biology screening services where tumoroid lines from different indications can be selected for the user's experiments of interest. The screening is optimized and then compatible with a number of different readouts to get a milestone report for findings and start to outsource this type of work when it's of interest to really take advantage of some of the lines that we have in-house and do these experiments quickly. So a typical immuno-oncology screening service estimate that it would take about 20 weeks to go from sourcing the cells from our validated patient-derived tumoroid lines across a number of indications, engineering them as needed to look at the readouts of interest, be that GFP or other reporters, expanding those lines along with, for example, CAR T or CAR NK cells, and then culturing both cell types and combining them in co-culture assays to monitor immune cell killing by apoptosis and potentially cytokine release profiling as well. Similar workflows can be done with compounds as well. So the tumoroid lines that we have derived in-house can be cultured, optimized for scale down screening assays, and then tested with different compounds of interest to look at cell killing and cell viability, often in a multiplex method, to see the efficacy of potential anti-cancer compounds with these physiologically relevant models. All of these services come with key deliverables, including milestone reporting that detail tumoroid culture, any characterization of engineered reporter tools, as well as assay results, materials, and methods. And this is done in an estimated turnaround time of 10 to 20 weeks with better estimates given as we know more details about potential projects. Finally, a number of tumoroid lines are available for research use and can be used by customers if they want to jump into these tumoroid cultures without having to derive or source tumoroid lines themselves. So across colorectal, lung, breast, and endometrial cancers, excuse me, we have several OncoPro tumoroid lines available through Cell Bio Services. These were all established as treatment naive lines and have been extensively characterized and validated for growth, stable growth in OncoPro medium, and can be purchased and used by customers to really get a head start on these tumoroid experiments with lines that we know work and that we know are representative of the original material from which they were derived. This can all be done in a larger ecosystem. We like to look at our 5 steps for tumoroid models, going from sourcing these lines to culturing them and characterizing them, engineering and performing assays using Thermo Fisher products, and really offering support throughout this pipeline, both for users doing these experiments in their own lab or for users interested in tumoroid services that can incorporate any of these methods. Happy to say that we've had these out to a number of customers, and we've received the following testimonials, often talking about how OncoPro Medium has allowed them to integrate tumoroid models into their research and development programs, really making it easier to use these models in their everyday use and accelerating development of these culture systems in their labs. Ordering information for OncoPro is here. This is ordered as one SKU when it comes with these four components of the complete kit through one order. Indication-specific growth factors are also available through either thermofisher.com or through Fisher Scientific. We recommend heat-stable FGF10 for these cultures, as it lets users use a lower concentration in their media than native FGF10. We typically use 10 nanograms per milliliter. And also listed here are the key auxiliary reagents for use with OncoPro. So for suspension culture, we use non-treated culture vessels. We typically will use Geltrex as our basement membrane extract and proaccutase to dissociate these cells for subculture, and recovery cell culture freezing media when we're banking cells for long-term cryostorage. Additionally, a number of training and support resources are available. This can be done through live training with our FAS team, as well as on-demand training with e-learns, quick start guides, and videos online. We've also got a growing library of application notes that go through different parts of this process, from tumoroid derivation to compound screening and extension to different indications as well. A lot of this is publicly available through R&D posters that have been presented at conferences and on-demand webinars that have gone into more details about various parts of this workflow. So with that, hopefully we've illustrated how OncoPro tumoroid culture medium simplifies tumoroid culture to make these physiologically relevant cancer models more accessible. Key takeaways are that it was formulated specifically for tumoroids to promote the long-term maintenance of these donor-specific cancer characteristics in a simplified method that does not involve Wnt, Noggin, R-spondin, or small molecule inhibitors. It's scalable and automation-friendly and compatible across a number of cancer indications and donors. I really designed with the goal of making it seamless to go into the downstream applications, into the experiments that customers are most interested in. And again, a number of services and control lines are available to accelerate the use of these models in researchers' experiments. So with that, thank you. I'll acknowledge the NCI PDMR and other donors for these lines that were used to procure some of the established tumoroid lines using results here. And again, thank you for your attention.